So good morning, class. Have you all received your midterm score?
Yes.
So this is a final result that I have added one bonus point to all of you.
Oh no.
If you have any questions, please send
One
me an
bonus
email
point.
as soon as possible. Or if not, I cannot change your midterm score.
Cộng thêm một điểm rồi à? Cộng thêm một điểm.
We go to the lesson number 9. Is about some very interesting activity happens with the cells, apoptosis, stem cells, and cancer cells. In the lesson about the cell cycles, I mentioned about when the cells damage, when the cell pass the checkpoint, when they have damaged cells or DNA damage, it will be fixed. And unfixable DNA damage contained inside the cells, it will put the cells go to die by the apoptosis. So what is the apoptosis cell and how it's important to the cells? Apoptosis, or they have another name, is programmed cell death or cell suicide. Is a generous and convenient way to remove the cell that's no longer be part of the organisms. During the cell development, there's some cell need to be deleted, or some kinds of abnormal cells can hurt the rest of the organism they survive, such as cells are infected with the virus or the cell carrying the DNA damage. And the cells in an adult organisms may be eliminated to maintain the balance and make ways for new cells or remove cell needs for temporary tasks. For example, during these development of the embryos, you know that in the embryo stage, we don't have toes or finger, and then here is the pictures of the embryonics in the mouse house. During these embryos genesis, there will be internal digital web, and when we cut this slide under the microscope, you can see that so many cells dies by apoptosis, and then the results of this process, the toes and the fingers will appear. So this process occurs in all sort of vertebrate species that have a finger or toes. And the interdigital cell death here is the oldest and the best studied model of the programmed cell death. Second growth of the apoptosis is can eliminate the infected or cancerous cells. Third, the cell cycles, there is checkpoint, and if they find some DNA damage, the P53 will stop the cell cycle. To fix DNA. If we cannot fix the cell containing the DNA damage, is leading to put the cell to apoptosis. And if the apoptosis do not occur, it's leading to cancer. So the cell with DNA damage, pre-cancerous cells, and the cell infected by the virus need to be removed, undergo the apoptosis. To put that kind of cell go to die to protect our bodies. The next function of the apoptosis is the keys of immune functions, and it plays a central role in the development and maintenance of the healthy immune system. And when the B cell and T cells are in the immune system first produced, they need to be tested to see if they react against our body own cells components. If yes, we put that size of immune cells go to apoptosis. If no, if it failed to put this kinds of cell go to apoptosis, it's leading to autoimmune conditions. Autoimmune conditions, autoimmune disease, immune disease meanings. Các bệnh tự miễn ạ. Can you give some examples of autoimmune disease? So we have the immune systems. It need produce some immune cell to protect our bodies. When the first produced, they need to be tested whether they can go to find our own body cells. And if they cannot recognize our body cells, it must be died. If you cannot put them down, it will lead to autoimmune disease or autoimmune conditions. Bạn nào bị bệnh lý không? Bạn nào có người thân bị viêm khớp dạng thấp không? Thấp khớp. Bạn nào nghe nói đến lupus ban đỏ nhỉ? Đó. This kind of disease is bệnh tự miễn ạ. Bởi vì sao ạ? Khi mà tế bào miễn dịch của mình được sinh ra thì nó phải được kiểm tra xem là tế bào miễn dịch của cơ thể mình có đi đánh bọn virus vi khuẩn không, hay là nó lại đi đánh chính các tế bào trong cơ thể. Và nếu nó không thể nhận biết được thế nào là tế bào trong cơ thể hay là virus và vi khuẩn thì nó sẽ phải chết theo chu trình apoptosis, phải tự tử. Và nếu mình không thể ép nó tự sát theo chu trình apoptosis thì nó đi tấn công tế bào trong cơ thể mình và gây ra autoimmune disease. Second process are the immune cells were born to protect our bodies of the pathogens like the virus and the bacteria. And when our body is invaded by the numerous of the bacteria, the immune cells will increase and we defend or successfully defend the bacteria, but the virus, or we defend the pathogens. When the pathogen is clear, the immune cells must be dyed by apoptosis to keep the balance, to maintain the homeostasis in the immune system. And there is a process inside our bodies, it's necrosis. Apoptosis is a process in the cell that the cell decide to kill themselves, go to suicide. both apodosis and under this phrase, cell damage can result in the necrosis and the different appearance than apodosis. And I have a table to compare them. Here's the necrosis of the cell dead by the juries. The cell will swells and bursts, and the inflammatory response will follow. However, in apodosis, these nucleus will be fragmentations to small pieces, and they will be packaged into small vesicle here, and it will be reused or to be engulfed by the other cells. So, in necrosis, inflammatory response, in apodosis, no inflammatories. Inflammatory phần viêm. Necrosis lies or you make an accident like you cut your fingers, and this point the fingers, all of the cells will be damaged by the physical stress, right? And it's causing VM, and for apodosis, nothing happens. Just the cells decide to kill themselves by separate into the small piece, packaged in the small vesicles, and they will fuse to the other cells. Another academic word, like apodosis, is the cell kill itself, like autolysis, an enzyme kill cells. It's for use when you make these terms or word. So how we can detect the apodosis? Here is the picture, the nucleus under the microscope. On the left, in the left, you can see the normal cells. They have very big nucleus. But on the right panel, you can see that the nucleus will be fragments into small pieces during the apodosis. And how we can detect them? We can isolate the total DNA, and we can run by electrophoresis. And you can see here, after 1, 2, 3, and 4 hours, the fragmentation of DNA will occur. So, in the first hours, DNA is conserved. Second hours, started to see some DNAs be fragments. In the hour after 3 hours, more DNAs will be fragmentations, and after 4 hours, you can see so many small pieces of DNA. So this is the pathways of the apodosis. All the activity inside the cells will be activated or will be stopped by receive some signal from the external environment. So like more signaling between the cells. First, we have two pathways. One is extrinsic pathway is effect from the outside environment. Second, intrinsic pathway. While the extrinsic pathway is start with the signal molecule binding to the receptor outside of cell membrane, and the receptors of the apodosis is trials or fast. So when the signaling molecule bind to the receptors, in the first step, it will activate the fast. Activates fast will leading to activated two kinds of enzymes, procaspase and procaspase 10 to be activated caspase and caspase 10. activated caspase-8 and caspase-10 will go to activate the bit into the T bit by cutting this part of the bit molecules, and this step 3. And in step number 4, T bit after activated T bits will go to activate 2 types of enzyme, is BA8 and BAKs. So start from this point, the extrinsic pathway goes with the intrinsic pathway. Our second intrinsic pathway intrinsic pathway to apolysis is they can trigger by stress or damage of the cells, like DNA damage or stress on ER, lack of oxygen or some metabolic stress. Here's the reasons. And they started, or in response to this damage, the cell will decide to continue existence might be generated or cause the organisms as a whole. Then they decide to kill themselves by activating. Here's BH3 only proteins. They are the class of proteins, they including several pro and anti-apolysis proteins. And by the next step, the BH3 only protein activates will lead to the BAX and BAK activated. So you can see the fourth step of the extrinsic pathway is equal to second step of the intrinsic pathway. So when the BAX and BAKs activate here, we call the condition we call the MOMP. MOMP is mitochondrial outer membrane permeabilities. It changes the permeabilities of the cell of the membranes of the mitochondria. Nó thay đổi cái tính thấm của tế bào tinh thể của tinh thể. And the MOMP is what we call the point of no return. So when our BAX and BAKs activates are leading to the MOMP stage, or the apoptosis will start, nothing to stop them, and we call the point of no return. So when the permeabilities of the mitochondria is changed, tính thấm của cái màng tinh thể mà bị thay đổi, it will release the cytochrome C. The cytochrome C plays important roles in the mitochondrial electron transport trains. So when they escape from the mitochondria, it will form a complex proteins called the apotosomes. Apotosomes, one is formed, they can turn the procaspase-9 into caspase-9, and they can trigger further changes through other cells. When the caspase-9 form, they can function to promote the apoptosis by activating the caspase-3 and caspase-7. Here, caspase-3 and caspase-7, and apoptosis happen. They will cut all the cell to be into the small piece and package them into the vesicle. And here's when caspase-3 and caspase-7 activated, they can start to break the cellular materials, break down here. The break down the cell, break down they can condense and cut the DNAs to be small pieces. So it's quite complicated for you. So there will be a videos about these this for you to easy to or more easy to imaginations.
làm sâu rồi. Trong đấy có cả mic, 3 cái mic.
So there is a problem with the audio, so I So what is hypothesis? Did the cell program start or dead, or the cells suicided? The cells will die following the programs. And during the embryo's genesis, here the formings of the finger or toes in the vertebrate by the function of the hypothesis. And also in the workings of these immune cells also. It's also worked by one caspase enzyme families. These will be cut on things inside the cell, cut. The cells will become small pieces. There is two pathways. Extrinsic pathway respond to the signals of the external of the cells, and intrinsic pathway is respond to the stress inside the cells. So here first they can react with the ligand, FAST ligands. When the FAST ligands bind to the FAST receptors, it will activate the FADD's proteins. Here's the activates of the FAST proteins will go to activate numbers of the caspase enzyme. First is the pro-caspase and caspase 10 to be activated. The second pathway is intrinsic pathway, where they respond to the stress inside the cells. And the mitochondria is involved in the hypothesis. In normal ways, the BCL2 and BCL8 will bind to the BAX and BAK proteins to inhibit the apoptosis. But when apoptosis happens, the BAX and BAL will be relieved. They will be inhibit and relieve the BAX and BAKs. BAX and BAKs will change the permeabilities of these mitochondria and relieve the cytosol Cs. Cytosol Cs relieve will forming the apoptosomes. Apoptosome will activate the caspase-9, and caspase-9 will activate the caspase-3 and caspase-7, and apoptosis happens. And this is the roles of the apoptosis, roles in the growth and development of the cells, and also in the balanced number of the cell and immune system. So you can see this video at home for more details, okay? The next process happens with the cells. We have the autophagy. Auto means cell, right? Auto means cell. And phages is come from the words phagomer, meaning to eat. Third, it's a process that the cells eat themselves. And this mechanism of these autophagy is the cell eats itself. They can digest molecules or organelles that are dummies or obsolete. Lỗi thời hay ít dùng đúng không ạ? So you can imagine that the lysosomes is one organ inside the cells. Will in the part with autophagy is essential for the cell to eat themselves, and the bonus in that its survival process can lead to the cell that work more efficiencies, or the cells can eating other part of the cells. How does work? The process of autophagy is related to the ATG proteins. It affects the cells. It makes the autophagy. These organelles inverse in biomembrane, sealing them inside a garbage package, and scientists we call them the autophagosomes. So the thing needs to be digested, forms in the package called the autophagosomes, and next they will fill with the lysosomes. And when they are too fused, there is content will be mixed, and the enzyme will break down the rabbits to recycle nutrition for use. And this all about the autophagy. It's some kinds of detoxification of the cells. How we can induce the autophagy? First is the fasting. This means that you can stop eating to certain times, and it deprives your bodies of nutrition, forcing it to reproduce a new cell component functions. Or second, calorie restrictions. It can decrease the numbers of energy units, calories, and your body consumed. So instead of depriving your bodies with calories, you can limit them. Here is limit. limits the calories. And second, and third, you can switch into the high-fat or low-carb diet, and this is commonly refers to a keto diet. Ăn chặn keto đúng không ạ? So many women like the keto diets, so they can reduce the weight and activate the autophagy in our bodies. So this type of diets, these are change the way the bodies burn the energies. So instead of burning carbohydrates or sugars for energies, they can burn the fat instead. Fat here. So these tricks can trigger the autophagy happen inside the cells. And the third is exercise. Exercise can stimulate process, or that's increase the activities of these proteins. So these protein in very narrow tissues, ATGs, and such as strengthening your skeletal muscles. And it can induce autophagy depend on the types of exercise that you are doing and depend on its intensity. So, what difference between autophagy and apoptosis? The apoptosis is will balance the number of cells in multicellular organisms, while autophagy is balance the energy source inside cells. And this causes cell stress, like starvation. The starvation of the cell can lead into the autophagy, while in apoptosis, some intracellular programs will put the cells go to kill themselves. Autophagy is the lysosome involved in this process. As you see in the previous slides, something needs to digest packets in the small vesicle, then they fuse with the lysosome and the enzyme in the lysosome will digest them. Whereas in the apoptosis, lysosomes are not involved. The exercise will lead excessive of the autophagy. Too much autophagy will lead to the cell death, whereas the excesses of the apoptosis lead to atrophies. And the autophagy allows cells to survive under the stress, where apoptosis does not allow the cell to survive. And this is one common thing between autophagy and apoptosis is nerve inflammation response. While with necrosis, inflammatory response happens. The next part of the lesson today, we study about the stem cells. What is the stem cells? Stem cells is the cells with potential to develop in many different types of cells in bodies. Stem cells, we call them the blank cells, the unspecialized cells. Proliferations, it can renew themselves for long periods of time, and differentiation meanwhile, they have potential to give rise to specialized cell types. Here is one stem cell. It can differentiate in muscle cell, fat cell, bone cell, blood cell, nerve cell, epithelial cells, immune cell, or sex cells. This is the plasticity of the cells. For example, from the hematopoietic stem cells, it can divide to generate more stem cells. Here, they can divide themselves. You can see these arrows. They can divide to make other stem cells, or they can make a precursor cell that can differentiate into immune cell like T lymphocyte, B lymphocyte, eosinophils, basophils, neutrophils, or some monocyte. This kind of monocyte can differentiate into osteoclast, các tế bào loãng xương. Or macrophage, đại thực bào, or platelet, tiểu cầu, or red blood cell, hồng cầu. From one stem cell, hematopoietic stem cell, they can live in the numbers of the blood cells inside our bodies. So it's the plasticity of the stem cells. So here's when one stem cell divides, its daughter cells can either become stem cells or they can differentiate into the precursor cells. Here's one divide, here is stem cells. So they can renew birth, or here is a precursor cell. It can differentiate, it can biệt hóa to adipose cells. We can divide the stem cells based on the source of origins. It is embryonic cell and the adult cells. Or based on different action of potentials, we have a totipotent, pluripotent, multipotent, and unipotent. Totipotent, tế bào gốc tổng năng. Tổng năng is totipotent. It means it can differentiate into any cell types in our bodies. Pluripotent là toàn năng. Multipotent, đa năng. So, totipotent it can differentiate in any cell types in our bodies. Pluripotent, it can differentiate in so many cell types in our bodies. Multipotent is less than cell types than pluripotent, and unipotent is just only one cell type. Example of unipotent, like you have skin cells, skin stem cells. It can produce only skin cell only. The source, based on the source of the stem cells, we have the core blood cells, or we have the umbilical cords on inside of our bodies. On the bone marrows, it have the hematopoietic stem cells. Here's two ways we can take these stem cells. First, stem cell from the in vitro fertilization from the IVS. When unused the frozen embryos, it can be through the waste, but we can produce the pluripotent stem cell from these kinds of embryo. The second, the patients, we can take the nucleus of the patient's cells, and with the unnucleated of the donor eggs, we can fuse together to make embryo stem cells. And from that, we can differentiate to form so many other cell types of the patient. And this is what kind of treatment in the future that we expect that we can do it in the laboratory. Or here the other ways, like we have the adult cells, or we can reprogram factors. We put the reprogramming factors inside the adult cells. It can re-induce the pluripotent stem cells, and from that we can differentiate in so many other cell types. Those are the first advance of the bone marrow transplant, or the first successful hematopoietic stem cell transplant in treatment of leukemia. Bệnh màu trắng ạ. Bring the Nobel Prize for Donald Thomas in 1990s. So this milestone involving the identical twins and with bone marrow taken from the healthy twins and given to the other who have the leukemia. And certain blood cells grow uncontrollably within the patient with bone marrows, and when we replace these treatment by the chemotherapies or radiance do not work very well, but we can actually put more hematopoietic stem cells back into the bone marrows of the patients, and then it will produce more normal blood cells for the patient again. So the bone marrow transplant is replace the damaged stem cell with the healthy cell.
Adult stem cells or tissue-specific kind of tissue in the body. A third kind of stem cells is called induced pluripotent stem cells. These are regular skin, fat, liver, or other cells that scientists have changed to behave like embryonic stem cells. Like embryonic stem cells, they choose to become any kind of cell in the body. While scientists and doctors hope to use all of these kinds of stem cells to create new tissue to heal your body, they can also use stem cells to help understand how the body works. Scientists can watch stem cells develop into tissue to understand the mechanisms that the body uses to create new tissue in a controlled and regulated way. Scientists hope that with more research, they can not only develop specialized medicine that is specific to your body, but also better understand how your body functions, both when it's healthy and when it's not.
And now we move to the next part of the lesson today is about the cancers. The cancers is basically a disease of the uncontrolled cell divisions, and I think the term of cancers is quite familiar with you. The cancers is multiplies in cultures without the growth factor. So when we culture the cells inside in the laboratories, if the normal cells, if it's the normal cell, we need to put the growth factors for the cell to divide. With the growth factor, the cell will divide, and when we forgot to add the growth factors, the cell will not divide, no divisions. However, with the cancer cells, when we culture the cell under the laboratories, we do not need to put the growth factors. They can divide by themselves, so by so many patches. So the cancer cells, we can call them that they are ignored signal. Let's call them to stop dividing. The cancer cells can replicate immortality. It means that they can divide many more times than normal cells. So in general, humans can grow only normal. Humans can grow 40 to 60 round. It depends on the cell types, but the cancer cells is immortality. They can divide so many times they want, and it's because of them they have a width. Telomerase. Remember the part of the chromosomes in the lesson about the chromosome? They have a telomerase. So to end chromosome, they have the telomeres. It will be shortened a little bit after the subdivisions, and with the help of telomerase, activate is more than 90% in the cancer cells. It activates. It will prolong the telomere. It maintains the length of the chromosome cells, it can keep the cancer cell divide so many times they want. Second, they can promote the growth of new blood vessel cells. We call the angiogenesis, 环境大脉啊。And the third, they gain the ability to migrate to other parts of the bodies, of the metastasis, the ganglion. These three characteristics of the cancer cells. So how cancer cell can develop? by numbers of mutation, series of mutation. So you can see that the mutation happens all the time in our bodies. But with only one mutation, it cannot lead to cancer. We do have the cancers when the series of mutations leading inside our bodies. So the first innate mutation inactivates a negative cell cycle regulator. Here's, for examples of negative cell cycle regulator. We have a p53, tumor suppressor cells. And the next mutation, they can overactivate the positive cell cycle regulators, and this is one example of the cyclin plus CDK. If we study the previous slide, it will be familiar with you. And next, by the first, third, and fourth additional mutations, cancer cell will form. And they might form benign tumors or malignant tumors, depends. Here is the process of the cancers. First step of mutation. Series of mutation will make a cancer cell. Second, these kinds of tumors will be growth, and the tumor cell lose the organization and they can proliferate quickly, and angiogenesis ensure. Angiogenesis quá trình tạo mạch đúng không? The cancer cells form the tumors, and the tumor becomes big tumor. When they become very big, so in the center of tumors, they cannot take the oxygen, they cannot take the nutrition, and they need to send the signal to our bodies, to the circulation system in our bodies. Okay, so I'm hungry, send signals, hungry, please feed me. Cells, blood vessels, we form. We call them these angiogenesis. And the next, intravasation. The malignant cells spread through the blood vessel and lymphatic system. And for the last, metastasis, the malignant cell become established in the distant organs. Phần này quan trọng, cô nói tiếng Việt. Quá trình hình thành ung thư như thế nào? Đầu tiên là phải có đột biến. Đột biến ở bất kỳ vị trí nào trong cơ thể. Nhưng mà một đột biến không thể hình thành được ung thư. Trong cơ thể chúng ta hàng ngày có hàng ngàn hàng triệu đột biến xảy ra, và cơ thể mình sẽ sửa chữa các đột biến đó. Nếu đột biến không được sửa chữa và nó gây ra một series number mutation, một hàng loạt các đột biến liên tiếp, nó mới hình thành nên tế bào ung thư. Ở bước thứ hai, khi một tế bào ung thư hình thành và nó sẽ grow uncontrollably, và mình không thể kiểm soát được quá trình phân chia của nó sẽ tạo thành gì ạ? Khối u. Tumor growth. Khi khối u phát triển đến một kích thước nào đó, khối u quá to, thì làm sao? Ở trong trung tâm của khối u, các tế bào ở trong trung tâm không thể nhận được dinh dưỡng và oxy để tiếp tục phát triển nữa. Nó sẽ tiến hành một quá trình angiogenesis, quá trình tạo mạch. Khối u sẽ tiết ra các ligand, các signaling molecules, nói chung là gửi thông tin đến cơ thể là tôi đói đấy, gọi là cho tôi ăn đi, và tế bào cơ thể nhận được thông tin như thế thì sẽ làm gì ạ? Tạo mạch, angiogenesis. Các mạch máu sẽ xâm nhập vào trung tâm của khối u và nuôi dưỡng các tế bào ung thư ở trong khối u. Tiếp tục bước 3 là intravasation. Các tế bào ung thư sẽ xâm lấn, quá trình xâm lấn malignant cells. Các tế bào ung thư ác tính sẽ bắt đầu xâm lấn các thành mạch và bắt đầu di chuyển vào trong mạch máu để di căn lung tung đi khắp nơi trong cơ thể. Bước thứ tư, bước này thì khỏi chữa luôn. Di căn. Các tế bào ung thư ác tính đi khắp nơi trong cơ thể và nó sẽ dừng lại ở một số nơi, tạo nên các khối u mới. Bước 1 chữa được, bước 2 tạm chữa được, bước 3 có thể chữa được, bước 4 thì vô phương cứu chữa không được. And about tumors. Start with the tumor growth. We have two types of tumors. Malignant tumors we call the cancer u ác tính. And the second type of tumors we call benign tumor, lành tính. Lành tính thì chữa được bằng surgeries, right? We can cut it out. So this is a characteristic of the benign tumor. It cannot spread to other parts of the bodies. It generally localized in the small site. Slow rate of divisions. These still have the function like the normal cells, and it do not break out the originating organs, and it can easily removed by the surgeries. So when a patient has benign tumors, we do not say that they have the cancers, right? Khi mà bệnh nhân mà có tế bào, có một khối u mà lành tính thì mình cũng không gọi là bệnh nhân ung thư nữa, bởi vì mình có thể remove by the surgery. Malignant tumors, cái này thì ung thư để ác tính rồi. It can invade or spread to other parts of the bodies. They have a very high rate of divisions, and they can spread by forming the metastasis. Again, metastasis, di căn. Cell can travel through the circulation, and it's very difficult to treat the malignant tumor. This is the frequency certain tissues become cancerous. Here is for example, respiration system, ung thư phổi, digestive system, ung thư dạ dày ruột, soft tissue, ung thư mô mềm, bone and joints, ung thư xương khớp, skin, breast, genital, bộ phận sinh dục, endocrines, nội tiết, lymphoma, ung thư hạch. Okay, you can see the frequencies of this kind of cancer. So you can see the digestive system, respiratory system, very high rate, right? Skin and breath, high rate. Genital system, very high rate. Urinary system, high rate. Our brains, very low. Heart, very low. Organs is quite low. While the digestive system, respiratory system, very high. High in skin, breath, genitals, urinaries, very high. So the questions, why there are differences between the rates of cancer in the different organs inside our body? Là do tỷ lệ ung thư ở các bộ phận trong cơ thể nhưng mà não và tim tại sao lại ít trường hợp như vậy? Okay, please.
I think it depends on the environment. Well, you see that respiratory or digestive system is higher because we tend to eat more, like nowadays we tend to eat more unhealthy foods, and all the other things that we digest are actually kind of like
Toxic.
that toxic, I think. And especially the respiratory system, we have air pollution everywhere, and also cigarette smokes and other emissions. So we tend to breathe more of that toxic substance and leads to more cancerous diseases. Yeah.
What about bread? Ung thư vú ạ?
Yes, breast. That also kind of due to our environment, like more toxic substance, and that also it also affect our uterus, and it makes it become like more opposed to toxic substance and more cancerous, I think.
We talk about the exposed to toxic substance. I think the bread, brains, and the heart is exposed to equal number of toxic substance because they expose only with the blood, right?
Yeah.
After blood go through the liver, they travel around our bodies. And the chains of these bread or heart or brains meet the toxic substance as equal. Well, I agree with their one hypothesis that you say that if with the lungs in the respiratory system, we expose to numerous of the toxic compound in the air, like in the tobaccos, air pollution, and in the digestive system, we expose to numerous of toxic compound in the food. But what about for the other part of our bodies? Why the rate of cancer is different? Still different.
I think it also relates to genetics.
Genetics?
Like a part percentage, but
genetics is one time. Okay, it's one reason that's in very next time that the cancers, like if in your families, if you have mothers or grandmothers have the breast cancer, it will increase the chance, increase the possibility that you will have cancers.
yeah.
Yes, please. Or one more reason about it.
I think it because of difference in rate, like the nervous system of the brain, they when they grow up, they not division into another cell. They not division anymore. But in the other system, like the tissue or skin, they will die division to another layer. So when cell division, they will reconstruct DNA, and that is part in the mutation.
So it's increase the mutation rate in
Yes.
this class. Okay, I agree. And two additional points, one for the girl and one for the boy. Thank you for your answers. So we have the difference or frequencies of the cancers is due to several kinds of reasons. I agree with the girls for these the the parts of our bodies that's exposed to the external environments like digestive systems and respiratory systems. We expose to a number of toxic compounds in the environment. So it's increase the rate of mutation, right? And the second reason, like the boy just says, is related to the rate of cell divisions. In the cell cycle lessons, I said that some cell time just goes into the Z0 state. And we have the heart cells, we have the neuron cells, and also we have the bone cells. So that's why the cells stay there, they don't divide, and it will slow down the rate of mutation in that point. So that's the reason why we have different frequencies of cancers. It's very rare when you meet the heart cancers or brain cancers. For brain cancer, it's not really the brain, it's just we call the covering membranes of the tissues, pineal glands, or some other parts of the brains, or from the glial cells of the tumor brain. Nào, ví dụ như là với ung thư xương, ung thư tim hoặc ung thư phổi, ung thư não rất ít gặp, và nếu mà gặp thì ví dụ như trong ung thư não thì các bạn không phải là gặp các tế bào thực sự là các tế bào thần kinh, mà thường là các tế bào thần kinh hệ đệm này, các tế bào tuyến yên này, hoặc là các tế bào tuyến tùng này, ở những cái tuyến mà nó vẫn hoạt động và nó phân chia nhiều ở hệ thần kinh, chứ không hoàn toàn là các u não mà từ các tế bào thần kinh. Bởi vì sao ạ? Các tế bào từ tim, từ xương, từ não thì nó nhảy sang giai đoạn nghỉ, giai đoạn G0 rất là nhiều. Nó không phân chia thì nó sẽ không có khả năng bị đột biến, không có các mutations, và như vậy thì sẽ không có ung thư, no cancer. So you can see the different rate of the cancer in different tissues. So regulator of these cancers. There is two types of the cell regulators. It may promote the difference of cancers. First, positive regulators, overactive of the positive regulators. Oncogenes become the oncogenes here. Cancer agents activate the normal cell, pro-oncogenes activated become oncogene, and this make the cancer cell. And second is negative regulator. We have the tumor suppressors. One famous example is P53. If P53 inactivates, it can lead in the cancer. Here is positive regulators, overactivated. Overactivated is a form of these proteins that are found in cancer cells. A growth factor receptors may send signals, even the growth factor is not there, and it will become overactivated. Overactivate form this gene we call the oncogenes. In the normal, they are pro-oncogenes, no activated. So here's when pro-oncogenes mutation activated become oncogenes, the cancer will form. You can see it's normal. We have a rat. The rat proteins will receive the signal from the growth factors outside of the cells. When they activate the rat, the cell will division and proliferation of the cell happens. No growth factors, no information from the outside of the cells, no masses, no division. But when the oncogene, when the rat activate the rat form pro-oncogene become the oncogenes. They receive no signal from the outside of the cells, but they still tell the cells, okay, you need to divide. So there is a problems with the genes right here. Second times, we have the tumor suppressor. Example in P53. About 50% of the cancer cell are the problems with the P53. Abnormal P53. So here, P53, when they are damaged, P53 will activate, they will bind to the DNAs. They will stop the cells at the G1 stage, at the checkpoint to fix the DNAs. They can activate all the mechanisms to repair DNAs. If the DNA is unfixable, it can trigger the cell into apoptosis. They will kill the cell. They will tell the cells, okay, you have a problem with your, we have a problem and you need to go to dying. And the cell will die by the activated of P53. So that's why we call P53 tumor suppressor. It works abnormally, leading to 50% of the cancers inside the cell. Here's non-function of this P53 is our DNA damage. The cell keep going, the cell divisions, and damage in DNAs, or we call mutation, will pass on the cell cycles and is leading to series of mutation which pass through these subdivisions. And by series of mutation, of course, we have cancer formed. And we have the videos about the cancers.
The human body is a complex and wondrous living machine. Like any machine, tumor cells to neighboring and distant parts of the body. Spread of tumors to distant locations is of great importance in cancer. About 90% of the deaths due to cancer involve tumors that have spread around the body. The movement of tumor cells to other parts of the body is known as metastasis. Metastasis is a complex process during which cancer cells break off of the original or primary tumor and move through the body to form tumors at new locations. From the point of view of a cancer cell, this is a dangerous and often unsuccessful process. A trip through the body is full of hazards that cause the death of most cells that begin the journey, even tough cancer cells. To begin the process, individual cells must break away from the tumor and invade nearby vessels. The cells crawl along the surface of other cells and the fibrous, stringy structure surrounding them, and then force their way in. Shown here is the invasion of the blood supply. Once inside a blood vessel, the cancer cells may perish from a variety of causes. Some cells die simply because they are unable to survive floating around in the bloodstream. Others may become damaged and die when they squeeze through tight spaces or bump into the walls of the blood vessels. Still, other migrating cells may be recognized and destroyed by cells of the immune system. How and where the migrating cells stop is different for different cancer types. Once the tumor cells are no longer moving, they can begin the process of forming a new tumor by leaving the blood vessel and beginning to reproduce in the new location. This does not always occur, and cells that have made it this far may still die or fail to divide. If the new environment is suitable, the newly arrived cell will begin to grow and a new tumor will develop. One way that the development of cancer is prevented is via the death of defective cells. A cell that becomes mutated or damaged will first attempt to repair the damage. If that is not possible, the cells will commit the cellular version of suicide. An orderly process called apoptosis leads to the breakdown of key cell parts and the death of the cell. Cancer cells lose this critical capability and will continue to divide. This can lead to the accumulation of cells that can become more and more abnormal. Due to the high rate at which cancer cells accumulate mutations, a tumor that originally started as a single abnormal cell is actually made up of many slightly different cells. They are all cancer cells and are similar to each other, but they may not all have the same sensitivity to any particular cancer drug or treatment. When this mixed bag of cells is exposed to a drug, most of the cells will probably die. Those cancer cells that are resistant to the treatment may reproduce to reform a new tumor. Importantly, this new tumor will be unlikely to respond to the same treatment. Their mutations make them invulnerable to that drug.
So one question. You know that the cancer cells that can to generate cancer cells, one is these metastasis or malignant cells. So to be a malignant cells or to be the cancer cell that can causing the metastasis, what could be the characteristics or what could be the type of the cancer cells? It's not easy to invade the blood vessel. It's not easy to travel around the blood and forming the new tumors at a new place. What could be the time of the cancer cell to have this power? Nào, đầu tiên là không dễ để có tế bào ung thư đúng không? Bởi một loạt các series of mutation, một loạt các đột biến khác nhau mới dẫn đến được một tế bào ung thư. Sau khi đã có một tế bào ung thư mà nó phân chia không ngừng nghỉ tạo thành một khối u, thì cái quá trình ác tính, cái quá trình di căn xảy ra hay không nó lại không hoàn toàn dễ dàng, đúng không ạ? Tế bào phải vượt qua thành mạch máu, đi vào trong máu dưới sự chịu rất nhiều áp lực từ áp lực dòng chảy của máu này, áp lực từ blood pressure, áp lực từ các tế bào miễn dịch thấy một cái tế bào đi lang thang ở trong máu thì nó cũng tiêu diệt luôn, và nó lại phải có sức mạnh để vượt qua thành mạch một lần nữa để xâm lấn ở những mô khác nhau. Từ đó nó lại hình thành nên tế bào ung thư ở các vùng khác nhau trên cơ thể. Thế thì cái loại tế bào nào có thể hình thành cái tế bào ung thư ác tính như thế? Tế bào phải có tính chất như thế nào mới có thể di căn được? Nó chiếm từ is occupy 1 to 3% of all cell in this human. This is only cells with the ability to generate, regenerate. Tie yourself. Loại nào được ạ? Yes, please.
My guess would be the liver itself, because liver has quite a high rate of cancer
is not quite correct. It's not relevant to these questions. Yes, please. Okay, so I agree with you. It should be progenitor or stem cells. Because why? When they travel to other tissues, they must have the ability to differentiate to other cell types, right? If they don't have the ability to differentiate in other cell types, it could be eliminated by the immune system. Nào, một đặc tính của tế bào ung thư ác tính nó phải là một progenitor của một stem cell. Vì sao ạ? Các bạn cứ tưởng tượng là ở tế bào ung thư gan đi đang travel đến phổi ạ. Tại phổi nó làm gì ạ? Tại phổi mà mọc lên một khối u của gan nó có vô lý không ạ? Lúc đó nó phải biệt hóa thành một loại tế bào, một dạng khối u ở trong phổi để tế bào miễn dịch không thể loại bỏ nó. Nên là it could be the cancer stem cells. To be the malignant. Sorry guys, it's all about the lesson today. You have a 50 minutes of break.
Okay, so we will play again to revise the previous lesson.
Play your game rồi. Play game mà chết đấy. Cái game này hơi chết đấy. Game này hơi chết đấy. Dương đâu? Dương thế này. Dương ở đây. Dương.
So we will stop again.
Oh my god. Đi đây. Đi luôn rồi. Signal. Cái này nghe quen. Signal. Sao nó nhanh thế? Sao nó nhanh thế? Xịt. Correct. And là kinase. What are three states of cell signing? Shit. không nằm ngủ yên. Mất tim. Lắc. Great. Everyone following time long distance. Nó là cái gì nhỉ? Quên tên. Rồi, Edo vẫn đầy đủ. Đây đồ xe. 63 người
Chiều lớp mình thi lý á.
Yêu sinh liều lý mà.
Yếu là sinh lý.
Yếu cả sinh lý. Rồi lại mời mấy top lên này.
Thôi rồi, hôm nay cô sẽ cho về sớm 5 phút, cô sẽ cho luôn điểm từ 1 đến 10 nhé.